Well, hello everyone, and welcome to today's webinar entitled Weed Management in an Herbicide-Resistant World. I'm Chris Boomsma, Director of Education here at the American Society of Agronomy. It is my privilege, as always with these webinars, to be your moderator and to kick it off here. So here we go today. All right, so as with all the other webinars in this particular series, I want to thank our sponsor, the Crop Protection Network. Their generous support of this webinar brings it to you free of charge, getting some quality CEUs, some great learning opportunities, and as I said, free. So thank you, Crop Protection Network, for not only this webinar, but the larger series. All right, moving on. Now, note today, as with all of our webinars, if you're having any issues, please reach out first to my colleague, Lacey Edwardson. You can see her email address and phone number on the screen at this time. She can address any issues you may have. Her phone number is, in case you cannot see the screen, 608-268-4953. Again, 608-268-4953. Note that this webinar, which is different, is only 30 minutes in length. So it's half a CEU in either pest management or professional means, depending on your certification. Trying a little bit of a different model here today, some quicker learning. We'll see how it goes. We will still have a question and answer period at the end of today's session. So if you have questions, submit them through the questions portal. I'll take them, pose them to the presenter in the last 5 minutes or so of today's presentation. So submit them anytime. Note that this training session, just like all the others, is being recorded. You will have access to the slides and the recording about 1 week after completion. Be watching for future webinars offered by the American Society of Agronomy in the coming months, including more webinars in this series. And you can find more about those webinars in this series by going to the link on your screen at this time. All right, we're going to keep moving forward here. Since we only have 30 minutes, I want to introduce today's speaker, Mandy Bish, who is an extension weed specialist at the University of Missouri. In this role, she focuses on helping producers integrate non-chemical methods with herbicides to achieve satisfactory weed control. She also currently serves as University of Missouri's IPM coordinator and co-hosts the podcast War Against Weeds. So listen to that. Prior to her current role, Mandy worked at the United States Department of Ag as a research plant molecular biologist, where she explored how soybean can better resist fungal pathogens. She has a bachelor's in plant science from the University of Missouri and a PhD in cell biology and molecular genetics from the University of Maryland. Thank you, Mandy, for being with us today. It's a privilege to have you here. All right, before Mandy takes it away with the presentation, though, we do have a simple request. If you could participate in these 3 poll questions, they're about to come up on your screen one at a time. It'd be much appreciated. She's going to gather some information for her IPM program to guide today's presentation and discussion. So here comes the first question for y'all. I'm going to launch it right now. You should see it up here on your screen. We'd like to get to about 70-75% participation. And so if you could please vote. Looks like that poll is distributing right now. And if you could go right ahead, I am now collecting responses. And we're going to see once we get about, again, 70% of you folks participating. We'll try to get this done in under 30 seconds, though. That way we can keep on moving on this webinar. All right, we've got some good responses coming in. Thank you to those who have voted. Just a little bit more and we'll be there. All right, very good. I am now going to close it down. Very good. We've reached our mark. I'm going to share the results both with the audience now and also with you, Mandy. Hopefully these results are helpful. That is the first question. All right, moving on to the second one now. Next question. Similar in some ways. Here it goes. It should be launching at this time. All right, looks like the responses are being captured now, so that's fantastic. Thank you, everyone, for submitting. We're almost all the way there already for a number of responses that we were looking for, so very much appreciated the rapid response this time. It's great. All right, we are there. So I'm going to close this poll down and share the results with everybody. So there you go. You should see it holding on your screen at this time. And that is poll 2. And now, the last poll. And here it comes. Your way again. Please vote on this last and final poll of the three before Mandy begins her presentation today. All right, very good, folks. We're almost there. Awesome. All right, thank you everybody for voting today. And I will once again share the results for this particular poll question. So there you go. Here are the results for the third poll. And Mandy, I want to welcome you now to the program. It is great to have you here. I am now going to stop sharing my screen and I want to make you presenter. And we really look forward to your presentation today. So when you're ready, Mandy, you just go right ahead and take it away. It's all yours.
All right, I am hopefully going to share my screen.
We'll wait a second. If you have an issue, maybe it's on my end with stopping the sharing of the poll.
Yeah, it's showing the sharing of the poll results is what it says.
All right, we'll hide that and we'll try to reshare it in a second. All right.
All right, are you seeing my presentation, Chris? Yes,
That's
I can
perfect.
see it.
You
All
are
right.
ready to go, Mandy.
Thank you, and thanks all of you for joining today, and also for taking the poll to give us some information. I'm going to talk at the end of this presentation briefly about soil health, and we start to consider how soil health may influence some of our weed management decisions. So I really appreciate your feedback on those poll questions. Again, as Chris said, I'm Mandy Bish. I'm an extension specialist at the University of Missouri. I work closely with Dr. Kevin Bradley, who runs the Missouri Weed Science Program and is our state extension weed specialist. Today, when I use the pronouns I or we, this is the team I'm likely referring to who does the work. And we're fortunate that we get to collaborate and have support from many different entities. The projects that I'm going to cover today have had support in some capacity or another from the programs listed at or the entities listed at the bottom of the screen. Before we get into the bulk of the presentation, I kind of just want to take a step back and remind us of why we're even investigating some of these non-chemical methods that I'm going to mention today. And this example is with water hemp and soybean. That's our driver in most of Missouri. But I think the message still applies to most of our weed management issues, and that is the status of our post-emergent options when it comes to broadleaf weeds and soybean. And so the study that I'm highlighting on this slide is a study that was sponsored by United Soybean Board, and we've worked with our colleagues at neighboring universities. And what we've done is we've gone out and we've collected water hemp populations from soybean fields at harvest. So water hemp that was still standing and had seed heads on it at harvest. And the color coding, and then we screened those populations with 2 different doses of glufosinate or 2 different doses of dicamba. So some of our popular post-emergent options. And the question was just, hey, are water hemp populations still responding as they should to these herbicides, or are we losing some sensitivity to these herbicides? And so the color coding on the map kind of summarizes the big picture results. Any county that is shaded in is a county where we had at least 1 water hemp population collected from that county. Now, the blue coding indicates that for each replicate that we screen for that population, so if we screened these populations with glufosinate, every time we screen them, we did not see any survivors from that water hemp population. So blue would suggest, hey, the herbicide is working as we expect. We aren't seeing any reduced sensitivity. Now, for those counties with populations in yellow, we did see in 1 rep or a couple where we had a few survivors to that herbicide. In other replicates, all the water hemp died. So those are kind of our questionable, hey, we could use a little more information to pick this apart, tease apart what this is, but they're questionable. And then for those with red, those are populations in which we did see some survivors every time we did the screen. So that would suggest what we probably already don't want to admit, but probably already know that we are losing sensitivity to some of our post-emergent options, okay? And so that kind of highlights the need for some of these, to look at these non-chemical approaches that we've been investigating. You know, glyphosate wasn't a silver bullet. Our post-emergent current options are not silver bullets. Likewise, the approaches I'm going to show today are also not silver bullets, okay? They have their places, but they also have their limitations. And so the goal is to think about this as integrated. You know, my goal today is not to make anybody an organic producer, although if you want to be an organic producer, that's fine. But how can we adapt some of these non-chemical methods with our current practices and extend the use of the herbicides, okay? So that's the goal and why we're doing the research that we're doing on these non-chemical methods. And so over the years, we've done we've done research on a few different non-chemical methods. Today, for the sake of time, I'm only going to talk about weed electrocution and at harvest grinding impact mills. But for more information, we have slideshows on our website, weedscience.missouri.edu. And I'm going to plug that website a couple times. So today I'm going to highlight some of our weed electrocution findings. I'm going to highlight some of our seed terminator findings. And there's more detail on those as well on our website. And then Missouri Weed Science YouTube channel, we have some videos on these topics. So weed electrocution, then I'm going to focus on seed terminator. And then at the end, I'm going to talk a little bit about some work we're starting with soil health. Because like I said at the beginning, I think, you know, we're going to get to the point where soil health is going to affect our weed management decision making. So weed electrocution. Haley Schreier is a graduate student in Dr. Bradley's program who's been managing this work for the last couple of years, and we are using the Weed Zapper, which is manufactured in Missouri. So we're supporting a Missouri business, which is nice. And basically, if you're not familiar with it, the generator is on the back of the tractor, and then there's a copper boom, and the copper boom is going to electrocute any plant that it comes in contact with. I mentioned previously that there are some limitations and some positives to these. Well, one limitation of this technology is that you're probably going to only be able to control weeds that emerge above the canopy, right? Or you risk electrocuting your crop. But one of the big perks of this technology that sets it apart from a lot of the things being studied is that you can actually target weeds before they set seed. So our goal with a lot of these non-chemical methods is, can we keep weed seed from getting into the soil? This is one technique that shows promise in that. And this is what it looks like from the cab. As you're going over it, you can see a little bit of spark there. Basically, our boom right now is, we have a 15-foot boom that we can adapt to make it 10 foot. I believe the commercially available, the largest one is 40 foot. So our goal right now is to test the efficacy of this tool, but there would need to be some adaptations made in order to make this commercially accessible or practical. And this is so this is giant ragweed, she's electrocuting, and this is what it looks like 24 hours after. So you can see on the right, those plants came into direct contact with that copper boom, whereas those on the left were not touched. So there is a pretty immediate, you know, if you want instant gratification on weed control, this might be the technique for you. But one question also about this is, what is it doing to the plant on the inside or physiologically? And so what we've been doing also, or what Haley's been doing, is taking sections of these plants and looking at them under the microscope. And so this is a giant ragweed plant that was not electrocuted. I apologize for the numbers in these pictures, just ignore those numbers. They just help us keep the plant with the microscope picture sample. So you have a non-electrocuted plant. Haley took a section of stem and took a picture under the microscope, and you can see the ducts are intact, the vascular bundles are intact. It's a pretty stem. So she came back after she electrocuted and collected plants that had been electrocuted and took a cross section from the stem. And interestingly, it's not like all of these compartments are just exploding inside the plant. You do see some browning or necrosis in the vascular bundles. And so this is just observational. A lot more data would be needed, but it seems like the signal is transferring up and down the plant through the vascular system potentially. So it was pretty fast too for 24 hours after. So when we look at this across different weed species, we see that the best visual control was generally observed when weeds were further along or in the reproductive stage. That's a generalized statement. There are some caveats, but in general, this is what we see. So along the x-axis, you have giant ragweed, horseweed, and water hemp, 3 different weeds. And then this is the control at 21 days after electrocution. So on this particular graph, a larger bar is better because it means more control. The timing of electrocution is shown in these individual color bars. So 30.5 centimeters is about 12 inches and 24 inches for the gray bar. So you can see in general, the better control, over 90% control, is achieved when these plants were targeted at the reproductive stage. And so I'm not showing the grass graphs today for the sake of time, but we do see a similar trend. One of the concerns with grasses is that the growing points are often lower than where the boom is sitting. And so we think better control is achieved when you have the growing points at or above where the boom comes in contact. Another study that we're doing with support from the North Central Soybean Research Program is we've been able to look at weed electrocution in different states, so taking it to different geographies and topographies and trying to learn what's happening there. And this project is led by grad student Jake Vaughn. And this is very preliminary data. We've only done it for one season now, but what we're starting to see, this is just a scatter plot, so not a lot of statistics to it. It's more observational. But as the pigweed plants are taller, the taller they are above the soybean canopy, the better control you get, which again would make sense. So along the x-axis, you have the pigweed height minus the soybean height. So the pigweed was 20 centimeters taller than the soybean here, 40 here. And as you get a taller difference, then your visual control rating is also tends to go up. Again, there are a few exceptions, but that is the overall trend, suggesting that that height difference between the weed and the canopy can influence the control with this. So I mentioned previously that one of the benefits of weed electrocution is that you can get to the plants before, it doesn't require seed being on the weeds. But what happens if you were to electrocute and seed heads had already started to form? Can electrocution affect that weed seed? Can it make it non-viable? And so to do this, Haley and a team of students cut through a lot of weed seed, and that's what we're looking at here on this image. You have water hemp seeds, which most of you are familiar with. They're very tiny that she's actually cut through. And then you can stain those embryos with a dye that turns pink if there is respiration occurring, so if the weed seed is viable. And so she has kind of 3 categories here. 1, there was no seed that formed following electrocution. 1, the weed seed was not viable. There's no pink staining of the embryo. And then some that were viable, okay? So that was what she was looking for, and she did this across many different weed species. And in these numbers, the higher percentage is better. So this is the percent of seed that was rendered non-viable once it was electrocuted. So common ragweed, 80% of that electrocuted seed was not viable compared to the non-electrocuted seed. And I apologize, it goes down to 54% of giant foxtail. So regardless, there's quite a variation there, but regardless of the weed seed, greater than 50% was rendered non-viable. So even if you were to electrocute after weed seed formed, you still have a chance of rendering some of that seed non-viable. So even though it goes back in the soil, it won't grow. So some highlights on electrocution. You can see instant gratification. There are instant results if you have good contact between the boom and the plant. This is a method that can reduce the number of viable seed produced from escaped weeds. So this again is not for that early season weed control, but going in for escapes and trying to really minimize the amount of weed seed that falls into the soil for subsequent years. There's probably many factors that influence the efficacy of this that we're still learning about, like that height difference I showed between the weed and the canopy. Soil moisture might play a role, soil type, different topographies. We still have a lot to learn. And there will definitely need to be modifications for this to be integrated into large-scale row crop production. If you have any interest in learning more about this, there's more detailed information on weedscience.missouri.edu under slideshows. Now I'm going to switch and focus on the impact mills, and this is work managed by a graduate student, Travis Winans. And basically the idea of the impact mills, if you're not familiar with them, is that it's a set of grinding mills that pulverize weed seed once it gets to the back of the combine. So it's separated from the chaff and then pulverized. And one of the challenges with this method is that the seed has to remain on the weed at harvest. So this is only for those weeds that produce seed and that seed remains intact on the plant at harvest. From work done previously by our group and many other groups, we know that the pig weeds make good candidates for this because once that seed is produced, it is retained for weeks after soybean maturity in general. And there are several groups studying this. And I will put a plug on this. This innovation started with a farmer idea in Australia. So they have some pretty big challenges with ryegrass, not cereal rye, but ryegrass resistance in wheat. And that's how this kind of whole idea evolved. And it was something that you pulled behind the combine. And now we're studying some integrated grinding mills that can fit in the back of our Class 8 combines here, okay? So again, the questions with the impact mills and this grinding is, you have to target the weeds with seed heads intact. And ultimately, can these impact mills reduce the number of viable weed seed that goes back to the soil? And I know I keep belaboring that point, but that's a really important thing to consider now. One of our goals has to be reducing the number of weed seed that return to the soil for subsequent years. So one question about this work is how much weed seed gets lost and never makes it to the back, to these grinding mills in the back of the combine. And so to study header loss and then what comes out in the chaff, we have these trays that you can see, they're in the circle right there, these silver trays. And basically this is showing the chaff measurements, but you would take these same trays, you put them in front of the combine, let the combine harvest. As soon as the header goes over the tray, then you stop and remove these trays, and that's how we're getting the measurements for header loss. Likewise, you put them behind, or you put them so that the chaff falls into them after the combine runs by, and that's how we're getting the chaff measurements. And so when we look at how much seed is lost, the water hemp number specifically, about 31% is lost when the plant comes in contact with the header. So 69% came out in the chaff. This is averaged across 7 site years, and it's pretty similar to what our colleagues at Iowa State have seen in some of their work. And so this is 69% came out in the chaff. So that means that 69% of the seed is a candidate for being ground in these impact mills in the back of the combine. Well, how do we determine how much seed gets into those impact mills and how much it gets passed through them and how much is being damaged? Unfortunately, there's only one way to do that, and you have to collect it. And so this is one of those not pretty jobs, trying to get that seed out, but it was successful. We were able to get it out. I will say, most of you are familiar with what chaff looks like when it comes out the back of the combine. But once that seed terminator is engaged or those grinding mills are engaged, that is very, very fine, much finer than sawdust that we were dealing with. So very messy job. So what percent of water hemp that actually made it into the seed terminator gets damaged? Well, this is just an image showing damaged versus non-damaged seed, okay? So the non-damaged water hemp seed, you can see that the seed coat is on tap. Whereas the damaged seed, you have some of the seed coat is missing. There are usually protrusions on this seed. And so the question is, how much of it looks like this is non-viable? So 95% of water hemp seed that exited the seed terminator was damaged, which is a pretty good number. If it gets to the seed terminator, it's likely going to be damaged. But not all of that weed seed that gets into the combine is going to make it into the seed terminator, right? There is still some that comes out of the chaff. So when you take that into consideration, about 74% of water hemp seed that exits the combine was damaged when the seed terminator was on. And so ultimately, that's good, but what does that look like in the soil, right? The goal of this is to reduce the number of weed seed in the soil over time. And so following the seed terminator runs in the field, came back the next spring and took soil samples and then determined how many water hemp were in each of those soil cores. And so all of these graphs are from different locations, and the black bar were taken from soil samples where the seed terminator or those impact mills were not turned on, so just normal harvesting. The gold bars are from plots where the seed terminator or those impact mills were engaged, okay? And these are water hemp counts. And what we found in general is that there is a trend, and you do start to see reduction in the number of water hemp seed coming up from those from the seed terminator plots. Okay. We also are thinking that this is not an instant gratification technique, and that it may take multiple years to start seeing the benefit of this if you have a really densely problematic field. We always have questions about combine performance, which is not my wheelhouse, but this is the summary of the combine performance as summarized by Travis. And fuel consumption is greater, the engine load is higher, and the speed was also a little bit slower. So just in summary of the impact mills, we do have header loss of the weed seed. So again, it's not going to be a silver bullet. That's why we have to think integrated. But most of the weed seed that made it into the seed terminator, those impact mills, was damaged. Another thing that I didn't have time to show today, but we do have data on, or I should say we have observations on. When we talk to our colleagues in other states in southern, like Arkansas, it seems like frost is going to play a role in how well this works. The earlier the frost, the better. Green tissue can be a problem for this method. And then, as I mentioned in a couple slides ago, when you have fields with high quantities of water hemp seed in the soil, it's likely going to take several consecutive seasons before you see substantial reductions. And again, I'm just giving you an overview of some of the highlights today in this 30-minute format, but we have more information, a lot more details at weedscience.missouri.edu in the slideshows. There's a whole slideshow dedicated to this, and there's a whole slideshow dedicated to weed electrocution there as well. And finally, I want to talk a little bit about weed management practices and soil health. The more we think about soil health, the more it seems that weed management practices are going to be influenced by our considerations of soil health. And so I do, again, thank you all for taking that survey earlier. And something that I'm sure that this audience can already appreciate, but that I try to just keep reminding, and I stole it from Larry Steckel at Tennessee, residual herbicides cannot be viewed as a Band-Aid. So in current weed management practices, residual herbicides are a must, regardless of what non-chemical methods we're gonna incorporate with them. And so, residual herbicides, soil-based herbicides make a good candidate for studying, well, what happens to the soil health when we have these residual herbicides. And so with the help of Syngenta, we started a project in 2021, just kind of starting an overview of, well, what's going on with the soil with different weed management tactics. And we included some different practices such as conventional tillage, no-till or cereal rye cover crop, soybean or fallow, and then we had some herbicides. And we looked at different intervals throughout the season to just see what herbicide concentrations were and to get an idea of the soil microbial biomass. So, do you have less or more bacteria in the soil after you have a residual herbicide application? Do you have less fungi and so forth? So we're looking at the living part of the soil, the microbial biomass in these particular assays. And so this is just an overview for the sake of time today, and it's still preliminary. We're still working through it, but one of the punchlines is that we really haven't seen a difference when we look at this end season between the plots where you had no herbicide versus those where you had residual herbicides applied. And if this trend continues, I think that's a really good that's really good as far as our options for residual herbicides. So what this graph shows is you have the different colors represent your different herbicide treatments, and the red is no herbicide was applied to those plots, no residual herbicide was applied to those plots. And so this is just the fungal biomass that accumulated 28 days after application. So you can see your non-treated fungal biomass, and then each of the herbicide treatments, and maybe mesotrione has a little more, but statistically they're all similar. So that's why we need multiple years of data too to see if there really are, if this is really different or just similar. And the same with our arbuscular mycorrhizae fungi. So these fungi are a different group, and they're the fungi that are kind of responsible for attaching to the plant roots and extending that network of roots to absorb more nutrients. So important class of fungi, and you can see that there's no difference again So again, this, I think no difference is a good result here, and we're going to continue this work on. Another question that we have about this is, well, we know that the mass of fungi is similar, and we know that the mass of the arbuscular mycorrhizal fungi are similar, but are the species the same? So we're kind of going to be able to do some sequencing to make sure that yes, we're seeing the same species, or no, we're seeing different species, and tease that apart a little further. And finally, one thing I wanted to include in this was just that, we had a weird season in Missouri this year. It was wet, 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 and then it was dry at the beginning of June, and we planted and we sprayed our residual herbicides. And there were some questions about, well, if you're doing something like this, you want to make sure that the herbicides were actually activated and available in the soil. And so this is water hemp emergence at the same time, where we looked at the soil microbial community. And it's a lot of bars, but like on the right are your no herbicide in the different environments or different practices, and then the different herbicide treatments. And on this one, it's the number of water hemp, so a smaller bar is better. So you can see that there is evidence that the herbicides were activated, and we also have the herbicide concentrations in the soil that suggest it. So no difference would be a good deal here. So going forward, soil health is likely going to have some influence on weed management decision making. I thought it was very interesting in your all's results that y'all, most everyone doesn't think weed electrocution would have an impact on the soil health. And we've had some questions about that, like would it or would it not? So that's great. And then, preliminary results from 2021 in Missouri suggest in season soil microbial biomass is similar between the herbicide treated and non-treated soils. But we do have so much more to learn. Environment, soil type, soil temperature, soil moisture are all likely going to influence the soil health properties. We already did the survey questions. That was my reminder for them. So I thank you for that. And two things to wrap up. Chris mentioned it at the beginning. The War Against Weeds podcast is a weekly podcast with Sarah Lancaster at Kansas State and Joe Eichele at North Dakota State. If podcasts are a way you like to receive information, we try to cover pretty relevant topics. Last week we talked with Greg Dahl about adjuvants, and this week it was with Don Refsel about herbicide formulations. We're going to have some discussion on how weed seed spreads in the next couple of weeks, and have drones and how to submit samples to a diagnostic clinic also in a couple of weeks. So just an opportunity, if podcast is the way you like to listen, and you can get those on Libsyn, or we're on Spotify still, iTunes, Google Play, your typical channels. And with that, I thank you all for your attention today. I'm happy to take questions, and I just want to highlight, you can find more information on these at weedscience.missouri.edu. You can also find us on Twitter at Show Me Weeds, and then we're on the Insta and Facebook, and we have the free ID Weeds app and a herbicide injury app that is 99 cents. So thanks again for your time. I appreciate it.
All right, thank you, Mandy. We are pretty much out of time today. So what I'm going to do, Mandy, is send you all the questions that were asked, and hopefully you can respond to these folks. We don't have a huge list. I think it should be manageable. And again, folks, we're trying this out, this new format, 30 minutes, trying to do some quicker learning, trying to fit it into your schedule, see how it goes. So let us know, actually. I'd love to hear your feedback, and we'll actually run a survey about this later. So Mandy, thank you for this great information. We do appreciate it. Folks, you can actually, if you have a question, you can see Mandy's email on the screen at this time. So you could reach out to her directly as well, and also Kevin. So thank you everyone for coming today. It feels a little weird wrapping up early, but I think it's good. And Mandy, again, thanks for your time. Thanks again to Crop Protection Network for their sponsorship of this event. Good quality education, free of charge. Come back for the remaining webinars. With that, have a great day, folks. Take care. Bye-bye.
Thanks.